I'd like to begin by thanking the organisers for inviting me here to give this talk. So I'm going to talk about mathematical modelling of angiogenesis, and I'm going to give two examples of the work that we've been doing. So one is on a PDE, a well-known PDE model, and that's going to be more theoretical work, which is on a phenomenon happening on one spatial scale. But of course, we know that angiogenesis, like most things in biology, is multi-scale. And what I want to do then is to present to you a preliminary work that we've been doing in terms of developing a multi-scale structure in which we can look at angiogenesis. Okay? And make sure I give proper credit to the collaborators. So Helen Byrne, many of you know, has been highly involved in this, along with Joe Pitt-Francis, who's done a lot of the computational work. Part of the work has been done with the drug company Roche, so collaborators there. Part has been done with colleagues in the Radiation Oncology Centre in Oxford, and then lots of collaborators, graduate students, and postdocs. Okay, so I think most of you know what angiogenesis is. Basically, it is a normal process that happens in the body. When you get an injury, new blood vessels form in order to repair the injury. And this is also what happens in cancer. And the formation of these new blood vessels is called angiogenesis. And the work I'm going to mention has been published with Samara Pillay, who was a graduate student of Helen and myself. And this paper here is about to appear. Okay, so here's a cartoon of angiogenesis. So basically what happens is you have a tumour cell. The tumour cell produces various growth factors. Now the blood vessel has got pericytes which sort of keep the blood vessel well insulated so the blood can flow along and doesn't leak out. And these are stabilized by a protein called Ang1. And what these growth factors do when they reach the blood vessel is they cause the cells to produce Ang2. And Ang2 interacts with Ang1, downregulates it, and the result of that is the pericytes move away. This allows the endothelial cells to break away. And they break away and they move up the gradient of VEGF, so they move towards the tumour. And they form blood vessels, and that's how you get angiogenesis. Okay? A lot of work, so you can see immediately here the multi-scale aspect of this, because you've got these proteins, Ang1 and Ang2, interacting with each other. You've got a tissue scale level, which is the VEGF moving from the tumour to the blood vessels. You've got cells moving, breaking out and moving and coming together. So you've got a huge number of scales there. And most of those individual parts have been modelled by many, many different people. Okay? Too many people to list, but lots of mathematical modelling has been done on every aspect of that problem. And one of those models has been on the model of the blood vessel, the endothelial cells moving towards the tumour. And this is the very well-known snail trail model that's been around for over 30 years. And the idea behind the snail trail model is that you've got tip cells that are like leaders that move towards the VEGF, move up the gradient of VEGF, and the cells behind them divide. And these are the stalk cells. So this is how you get the growth of the blood vessel. Okay? And this was first put forward actually for fungal colony growth in 1982 by Leah Keshet, and then adapted to angiogenesis by Baldwin and McElwain in 1985, and then Byrne and Chaplin, two people whom I'm sure you all know very well, and good friends and collaborators. And this model here is a model that we've been using a lot. So the general idea is that you've got these tip cells that move along, and they leave behind, and behind them are these stalk cells growing. So how are you going to form a network? Well, you can form a network from these two things coming together like this, or coming together like this. And the process of coming together is called anastomosis. So this would be tip-to-tip anastomosis, and this would be tip-to-stalk anastomosis. And that's how you get a network and a circulation. So the first thing you should see about this is how this is really dimensionally dependent. In 2D, two lines will almost always meet. In 3D, two lines will almost always miss. Most of the simulations of this work, most of the modeling of this has been done in 1 and 2 dimensions. So an artifact of doing that in 1 or 2 dimensions is you get networks to form. And you wouldn't get that in 3D. So there's an issue here of if you're in 3D, how do these things meet? And that's something we've been looking at, but I won't have time to go into that today, but happy to talk to people about that. And this is the snail trail model, which will be familiar to many of you. And this is looking at 1 dimension, okay? So N is a tip cell density, and C is the growth factor, the chemical, and E are the stalk cells. So the first bit says diffusion, movement up the gradient of chemical, so that's the chemotaxis, and then more tip cells form as the VEGF density increases. That's observed experimentally. And then the beta times E is if a tip cell meets a stalk cell, then the tip cell gets lost and it's no longer a tip. And the N squared term is if 2 tip cells meet. They get lost because they come together, okay? Now you can write that first equation, the first part, as the divergence of a flux. So if the flux, the movement, is what's causing the cells behind to divide, then the flux is the source term for the new cells that are forming. And that's the second equation. With the 1 over h there, it's a scaling thing, not to worry about. So that's the equation that many of us have been working with, including us. We've been using that a lot. And so the question I want to ask is the following. How good is the snail trail model as a model of the snail trail? Okay, so forget about angiogenesis. Just think about that phenomenological description I had of a cell moving along, causing division behind it, and moves up a gradient. How do you model that? That's the question. And we all use the snail trail model, and the question is the snail trail model a good model of the snail trail? And the answer is no, but yes. Okay? So if you want to sleep, you can sleep now and get up when the answer is yes, and you'll be very happy. If you want to be sort of panicked for a while, then keep awake. Okay? So what we're going to do, since all we want to think about is the movement, we want to make the thing as simple as possible. So we're going to make the assumption that the chemical is just constant. not constant, I mean, is just a linear gradient. And it's a fixed linear gradient, and we're going to have cells moving up that gradient, okay? And what we're going to do then is we're going to have a square lattice, and then we're going to have the probability of moving. So if it was just random movement, and you're in a square lattice, moving to nearest neighbors, von Neumann neighborhood, you have 4 possibilities. So there's a quarter probability of moving to each one of them. But now this is going to be biased by some function g, which would be related to the gradient of the chemical, okay? And the gradient of the chemical, just straightforward gradient term, where we're in 2 dimensions. So having said 2 dimensions is really bad, I'm going to work in 2 dimensions. So at the ith, comma j part, the x derivative is simply c at I plus 1 minus c at I minus 1, etc. Okay? So what we're going to have here is if 2 tip cells coincide, they form, 2 tip cells go like this, the tip cells disappear, and you get an endothelial cell. So that's TC plus TC gives you EC. If TC hits an EC, TC disappears, and you get another EC. So these are the rules for the individual-based model, and the rules that other people have used. There's nothing new about this. Okay? How do new cells form? How do new tip cells form? We're just going to say that they branch perpendicular to each other. And the probability of that branching is going to be proportional to the VEGF concentration. And that's simply because of the experimental literature that suggests you get more branching as you get closer to the tumor, presumably because the VEGF concentration is high. Okay? And so just look at the top there. These are sort of pictures you get. So now we have got the gradient. So we've got the tumor is on your right-hand side, and the blood vessel is on the left-hand side, and the cells are just moving up the gradient. Okay? And we want to compare that with the snail trail model. And what the snail trail model does is essentially says this is all in 1 dimension. So essentially it is collapsing that all down to 1 dimension. In other words, it's integrating along the y-axis. Okay? So what we will do to compare it with the snail trail model is take ensemble averages. So we run the simulation, and then we average in the y direction in order to get the x dynamics, okay? And if we go back here and look at the black thing, that's the averaged in 1 dimension, okay? Right, so how do we write a mathematical, a model of this that we want to try and get a PDE model? Well, it's a very, very simple thing. We just say that if you look at the occupancy at I, j, at the site I, j, and you want to find out how can I change the occupancy of that point for a certain cell type, well, it'll be the probability that there was a cell type next to it and moved into it, or there was a cell there that moved out of it. And then that's that term up there. This we're talking about the tips. And then if 2 tips coincide with each other, you will lose. So that's tip-to-tip anastomosis, and then tip-to-sprout anastomosis. So by just going through and doing the bookwork, you can write all these things down, and then you do the obvious thing, expand everything in the Taylor series, and keep the lowest order terms. And when you do that, and that you do that for both, you end up getting this set of equations here. Okay? So N is now the tip cell density, and E is the stalk cell density. So one important thing to notice here is that typically when you do the limit, so you have a mu, you have an h squared over 4 tau, so h is the spacing of the x, y, the spatial going from one box to another box. Delta t, which is here we call tau, is the time step, and then typically you say, let h squared over tau, what's the limit of that as h goes to 0 and tau goes to 0? Let's assume that's fixed and that's your diffusion. That's how you get the diffusion term. If we do that in this case, there's an issue because we also have terms of the form p over tau. And as tau goes to 0, that goes to infinity. So there's a mathematical issue there that at some point we need to get around to dealing with, but we're going to follow what people have done in the literature, and we're just going to leave that in there because in our simulations, we actually don't take delta t going to 0, we just take it to be very small. But there's a mathematical thing there that needs to be done, so we need to do that, okay? The boundary conditions are as expected, so they're all very boring, just zero flux, so we'll ignore them. And now, to get to one dimension, we integrate in the y direction, and we need to use a mean field approximation, and when we do all of that, this is what we end up with. Okay, so at the top you see the model we got, and at the bottom you see the snail trail model that we've all been using. And let's look at the very top, we've got the diffusion term and the chemotaxis term multiplied by something. Compare that with the snail trail model where the same term is multiplied by 1. Okay, so those two terms are different. Now, AN is a probability, so it's less than or equal to 1. AE is a probability, so it's less than or equal to 1. We've normalized the densities to lie between 0 and 1. So if you look at that bracketed term at the very top there, the extra terms in there, we could convince ourselves that they are very small, because they're multiplying, two terms multiplying together that are less than 1. Okay? But notice, there is the possibility that that term could be negative. So you could get a negative diffusion coming in. Okay? Which is not something that we thought would come out of this. Right? Now, if you look at the other terms, the kinetic terms, there's an n squared, an ne, and a cn, just like the one below. So if you look at the equation for n, for our model and for the snail trail model, they're roughly speaking the same. So that's fantastic. Okay? Now let's look at the equations for E. Look at our equation for E and look at the snail trail equation for E. They're not even the same type of equation. One's hyperbolic, one's parabolic. They're completely different. Okay, and that's not what we expected. Right?
So what about 3 dimensions?
Oh, we're not worried about 3. Well,
you do the same.
You could do it in, yeah. So in fact, when we're doing it here, if you do it in 1 dimension, then if you go to higher dimensions, you have to add extra factors in to account for the dimensionality. So for example, going from 1D to 2D, you have to add a factor of 2 in. Because it is a very important point, because in the snail trail model, if you're modeling going in this direction, suppose you did your sprouting and you went up like this, then in the snail trail model, that would say there's no increase in density because you project that back down to here. But in fact, you've created quite a big blood vessel there. So the models will underestimate the densities of things. Yeah. So now we've got three models. We've got the cellular automaton model, which is the true snail trail. We've got the snail trail model, which is the snail trail model that we've been using. And we've got this new model. And what I've just told you is that this new model is completely different to the model we've all been using. Okay, so now let's compare them. So black is going to be the solution, like the true solution. Red is going to be the old model. Blue is going to be the new model. Okay, now we go back to this picture, and in the second row, looks like there is just one line. Okay, there are actually three lines there. One of them's black, one of them's blue, and one of them's red. They sit right on top of each other. And the first one is the tip. The second one is the stalk. So both the snail trail model and this new model agree perfectly in describing the movement, the actual result, and you can't tell the difference between them. So that's a very funny thing. Two PDEs, completely different to each other, not even the same type of PDE, giving precisely the same answer. Very strange thing. Okay? Next one, roughly speaking, the same as well. Okay? So then we started looking at these models. We asked the question, what happens if there's no anastomosis? Total agreement. What happens if we have tip-to-tip anastomosis? Total agreement. I mean, there's a little bit of change between, you can see now a little bit of difference between the models, but by and large they're the same. Okay, so there's two things here. One thing is two very different PDEs giving you the same answer. There's the second thing is that our new model was much more complicated than the snail trail model. Snail trail model was basically linear. Our model isn't. So if the two models are giving you the same result and you've got a choice between linear or non-linear, what are you going to pick? Pick the linear all the time. Okay?
But would they give the same as a mathematical result?
We're working on that at the minute. We've got some ideas, but we haven't worked them through yet. Yeah, we're working on that at the minute. We haven't got it through yet. Yeah. So here's an example where we allow tip-to-tip anastomosis and tip-to-sprout anastomosis. And what you see here is the new model and the old model both agree with each other perfectly, and they're both totally wrong. Okay, the actual answer, the black line, which you can't see, is 0. Okay? So these models, the two models, the new model and the old model, agree all the time, whether the answer is right or wrong. Okay? So what we've done here is we'll say, well, okay, the way we got these answers was we did that limiting case, the asymptotics, to get our parameters. But instead of that, we could have just said, right, think of the simulation of the cellular automaton as being data. Can we fit these models to the data? And yes, we can, and you can't tell the difference between them. Okay? So what we've done here is develop this sort of systematic model for doing the snail trail. And what we found is a number of things we've been able to use. So you might ask the question, wasn't this a complete waste of time? You ended up getting this new model, it's the same as the old model. Well, in developing this new model, we came up with some implicit assumptions that we'd all made with the old model, and because they were implicit, we hadn't made them explicit. One thing is, there's a mean field assumption in the old model, and therefore, if you have a situation where the mean field doesn't work, where the system isn't well mixed, then neither of these models will work. So here's a question then, that could you, by looking at a network, angiogenesis network, would you be able to say a mean field model will work for that, or a mean field model won't work for that? We don't know. What we've also found out in this is that to get these models to work properly, you have to not allow self-loops to form. So in the cellular automaton model, the cell could sort of come around and do something like this. And if that happens, neither of these models work. So an implicit assumption in the snail trail model was that self-loops don't form. And that becomes apparent from this. So the conclusion about this is, how good is the snail trail model, the model of the snail trail? The answer is pretty good. But the issues are, and relating to what Avner asked, we have started doing some preliminary analysis of looking at these two different sets of models, and if we go into a traveling wave coordinate system, we can actually show that under certain circumstances, mathematically, we can show that they're equivalent. So we're still in the process of doing that. The other thing we can do, which I'm not showing here, but is in Samara's thesis, is we did a huge parameter space analysis, and we found parameter regimes in which the models do disagree. So we found a parameter regime where the snail trail model that we've all been using is not correct. But the question is that parameter regime a physiologically relevant parameter regime? And another question is, can we, by looking at that parameter regime, be able to say, when we look at a network, just by looking at statistics of the network, will the snail trail model work, or do we need to use the new model? Because given the choice, we want to use the snail trail model because it's linear and it's been well studied. Okay, and that's work that's still in progress. Okay, so now for the last few minutes, I want to go on to talk about multi-scale model, and I'm not going to go into very much detail here because of the time, but this is work, and this is concerning the cornea micropocket assay. So just a warning here, in that the next picture I'm going to show is a picture of an eye, and I know some people get a bit weird over eyes, so if you don't want to, so close your eyes if you don't want to see an eye, okay? So what they do is, this is the cornea, and what people tend to do, and this is not a human cornea, this is a rabbit eye. That green thing can be a pellet or it can be a tumor. You stick it in there, and what will happen will be that the blood vessels in the eye, in the cornea, which are around the outside of the cornea, there's no blood vessels in the cornea, it's all on the outside. So when you start to see blood vessels in the cornea, they have come because of what you stuck in the eye. Okay? So if you stick a tumor in there, and then you look at, maybe you can't see so well, and it's pity this isn't working because I can't really, oh, it's not working now. So here, this was where normally the blood vessels would be, and now it's gone up a bit. This is an artifact, the person who took the photograph, okay? See, now it's gone up to here. This is all tumor-induced angiogenesis. At day 5, it's gone to here, tumor-induced angiogenesis. So what the drug companies do is they would maybe put some growth factor here and see what happens, or maybe they put some anti-angiogenesis drug somewhere and see what happens, and then they compare. And then the comparison will tell you how good your drug is. So for example, if you find that with your tumor, you get this amount of growth, but then if you put an anti-angiogenesis drug here and you got this amount of growth, then you would say, well, that drug worked in some way, try a different drug, see how far the front goes, and then compare the two drugs, okay? So we want to do a mathematical model of this. So the first thing we did is to get data, is a bit of image analysis. So we take this here, image it, segment it, and then we're going to end up getting a one-dimensional model. And so what we do is we simply think of the dimension as going from the limbus on the outside to the place here where the pellet is. We think of it as being one dimension, so we integrate in this direction here and get something like that, which is the traveling wave of angiogenesis, okay? Now, this I mentioned at the beginning about all the interaction between the Ang1 and Ang2. We put that into multi-scale model. The model has tip density, vessel, well, we've called it here the limbal density, the stalk density, VEGF-A, and then they also look at something called the basic fibroblastic growth factor. We have that in it as well. This is how these two guys work. And we have the growth pellet, the growth factors are put in the pellet. There's dynamics of growth in the pellet. We put all that into the model. So basically, this is the opposite extreme of the first model. The first model was clear away all the complexities. Let's just have cells moving up a chemical gradient. This one is put in every single complexity we can think of within reason and write down a mathematical model. So this model will have the different cell types, the tip cells, the stalk cells, the blood vessel cells that due to Ang1 and Ang2 interaction broke away to form the source. It'll have the dynamics of Ang1 and Ang2, and it will have the dynamics of these two different growth factors in terms of how they influence the blood vessel, the cell movement, and how they interact with each other, okay? Now, come to think of it, I should have maybe had a warning sign for this next thing here. I'm not going to go through the detail of all of this. It's basically a big set of reaction diffusion equations, okay? So once you read a bit of the literature, write down equations, okay? So that's not really important. Model, continue, okay, parameters, millions and millions of parameters, okay, of which one or two you can find in the literature, and usually that's meaningless anyway, because you do the experiment on a different day at a different time with a different animal, with a different person in a different country, you get a completely different answer. So they're all a bit of a fiction anyway, parameters, okay? So what we can do then is we take the imaging data, and this was done for 9 mice for VEGF-A, 10 mice for the FGF, And then basically the green line is the average of the data at day 3 and at day 5. And the red line is basically the error bar, okay? Now, since we've got several million parameters, we can easily fit the data, okay? Dotted line data, okay? Right, you could fit that with 3 parameters probably. But then we can start doing things, like we can ask the question, what would happen if you did applied anti-B FGF and anti-VEGF? Would there be a synergy there? What would happen if you just did anti-VEGF? What would happen if you just did anti-FGF? So we can play with things like this, okay? And of course, we have to be very careful how we interpret the answers because of the huge parameter space we have. But now, and so I don't want to spend a lot of time in saying the answers of that, simply because there's this issue of the parameter values. But what I've got to, the issue I've got to bring up here is, how do we judge, how do we measure our data or model against the data, okay? So typically the state of the art at the moment is tumor angiogenesis network is an erratic mess. Here's my mathematical model. It produces an erratic mess. I've got a model of tumor angiogenesis, okay? How do you compare the two of them? What metric should you use? We still don't know. If you were to model human limb development, digit development, the number 5 is probably quite an important thing, 5. You would want your model, if your model predicted that every human should have 11 fingers, then there's something wrong with your model, okay? How would we know that my erratic mess is worse than your erratic mess in terms of comparing it to tumor angiogenesis, which is an erratic mess? Don't know, right? So that's an issue. How do we get summary statistics? That's one of the challenges in this field. And we haven't been able to tackle this because we didn't have the imaging. Now we do have the imaging, and you can, in space and in time, follow the imaging of the same vasculature. It's not like you had to kill the animal, stain the vasculature, and that's you had your one data point. You can now follow it. What are the statistics that we need to extract? I don't know the answer. What we did in our model, because it's one dimension, and it's roughly speaking a wave, was we said, let's take the maximum amplitude as a statistic. Let's take, so where's the front of this wave? If here's a wave, is this the front of it? Is this the front? Is this the front? Is this the front? Is this the front? What we said was, we'll take half the maximum amplitude and we'll call that the front, okay? Now remember I told you how do you use these pellet experiments? Well, you do different experiments, so you put different things in the pellet, and you basically see how sensitive the front is to the different pellet. And so if you put in pellet A and pellet B, and you find that the front in pellet B moved a lot quicker, moved a lot further than the front in pellet A, then that would tell you that in pellet B, you've got something that is better at angiogenesis. Okay? That's basically how you use this. So then we thought, since we've got these million parameters, and parameters are telling you something like growth rate, death rate, blah, 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 let's do that experiment. And let's find out to which parameter is the front most sensitive. So this might tell us what you're actually measuring when you do that experiment. So that's what we did. And now this is going to be a big letdown, let me warn you. You can't read this. This is sensitivity analysis, and you see here, basically, the bigger this line is, the more sensitive that parameter is. The model is most sensitive to that parameter. So here, using all these different statistics, maximum amplitude, location of vascular front, etc., in each case we find, and this is tumor density, in each case we find there's one parameter to which the dynamics is most sensitive. In other words, there's one parameter that if you change that ever so slightly, you will see a huge change in the dynamics of the vasculature. So fantastic. We've now figured out, when you do this experiment, what is the property of the pellet and the stuff in the pellet, what is the property that made you get more angiogenesis or made you get less angiogenesis? And the answer is, and I told you this was going to be very disappointing, where you place the pellet. So this model is saying to you that if you do two experiments with chemical A in pellet A, chemical B in pellet B, and you notice a difference, the difference could be that you place the pellet in a slightly different way and have nothing to do with what was in A and what was in B. So this is saying to our collaborators, it's basically a question of experimental design. When you do this experiment, make sure you put the pellet in exactly the same place. Now that's very easy for us, or for me, naive, totally impractical mathematician, to say. It's another matter to try and do that experiment. So the conclusion of this model is a bit worrying, really, in terms of what is your experiment actually measuring. Okay, and that's the key result to come out of here. We also got some results by playing around as to how the different growth factors might be interacting with each other. I don't have time to go into that detail. The last thing I want to point out is that in these models, there's no flow, there's no blood flow. But with our computer people, what we've done is made something, so there's something called CHASTE, which is software developed by my colleagues, computational colleagues, which stands for cancer, heart, and soft tissue environment. And what we've done here is made something called microvessel CHASTE. And within microvessel CHASTE, we have developed these types of models that I mentioned about, but put flow in them. And then what we've done is use those models to try and see, first of all, remember I talked about the cornea, and then I considered that as being one dimension. So I ignored 2 dimensions and I ignored curvature. Does that make any difference? So what we've done within this is to ask the question, with the standard modeling assumptions that people make, let's compare them, because nobody's done that. Do they make any difference? So that's what we've done in these papers here. And we've asked questions like, well, why would you want to know the angiogenesis? You'd want to know that because you'd want to know the oxygen delivery, because you'd maybe want to know the efficacy of chemotherapy and radiotherapy, and some chemotherapies' efficacy is enhanced by oxygen delivery. Radiotherapy is enhanced by oxygen delivery. You might want to know the oxygen delivery, the oxygen dynamics. And the question we want to ask is, different models make different assumptions, give different answers. Shouldn't we test those things? Now, we always complain about the fact that biologists, they get different answers when they measure parameters, okay? And we are rude about that to biologists. Well, if you think about it, they're dealing with living systems. What is the definition of a living system? It's something that responds to its environment. So of course, it's going to behave differently, depending on what time you do the experiment, where you do the experiment, how you do the experiment. Of course, it's going to be different. So it's very hard for biologists to really compare these things. Let's look at ourselves. We all have different models. We all make different assumptions. We could test and compare all our models if we wanted to. That's much easier to do than ask an experimentalist to compare the model and tell me the value of the parameter. Do we do it? Not really. Okay? And that's what, with this microvessel chaste, that's the sort of thing that we're trying to do. And with chaste itself, we're trying to look at, in multi-scale models, let's take different people's multi-scale models, let's compare them. Okay? And we've been having this discussion at lunch and dinner, etc. Of course, when you do that, it gets rejected, because it's not new science. What's the point of doing something useful if it's not new? Okay, do something new that's not useful, get published in a big journal. Do something that's useful but not new, and you have to fight for several years with reviewers. Okay? But you win in the end. You get them published in the end. Okay, so I'd like to end there. Thank the funding organisers, the people who funded this research, and thank you for your attention.
Questions or comments?
So I think I think you are doing a misrepresentation of yourself. Let me strengthen what you said. Number one, you did not have millions of parameters. I had less than 1 million, so you certainly had less than that. Number two, I know you know many of the parameters, all the half-life and all of these are already established. Yeah. Also some biochemical reactions, we know that thing. There are some parameters we don't know, you don't know, but you estimate order of magnitude, and you make some assumptions. And then once you do this, then sensitivity analysis makes sense because you are changing it within the order of magnitude. Yeah. So that's okay. Yeah. So I think I don't want to give these young people behind you think that, just any parameters. No.
Yeah, so that's a very, very important point, Avner. So, I mean, you couldn't read the slide, but there were parameter values in for virtually all the parameters there, and you're absolutely right. There were order of magnitude parameters there. And what that, and so in our sensitivity analysis, we just didn't pick any old random point in space and then do the sensitivity analysis. We picked values that, roughly speaking, people thought were correct. And then, of course, the real power of mathematical modeling in that respect, by doing a sensitivity analysis, is if you figure out to which parameter the model is very sensitive, then that's where you can inform the experimentalist, because you can say to the experimentalist, if you want to spend energy on measuring a parameter, measure this one really accurately. These other ones, even if you're out by 2 orders of magnitude, it doesn't make any difference. So you're absolutely right, yeah.
And one thing, what would be the second parameter after the location of the pallets out of the million, which would still show sensitivity? Just
I can't remember, because see what you see here, so this one is the front, this is the maximum, and this is the density. And what you can see here is that this parameter, the distance, in all of them is the most sensitive. The other parameters, see, this is very sensitive here. Actually, it's a bit sensitive here and here, but a lot of the other ones, there's a little sensitivity in one of them, but not in the other one. So the length was the only one that was most sensitive across.
There was not an obvious second winner.
Not an obvious second choice. Yeah. Yeah.
So the sensitivity analysis usually depends on the set point for
yeah.
So did you check how sensitive is your sensitivity analysis to the set point?
No, so that's another good point. So what we did do was, as I mentioned, relating to Avner's question, we took from the literature, from talking to our experimentalists, that set point where, roughly speaking, the parameters seem to be. And the problem with this is a computational problem, of course, because if you take different points, you can do the sensitivity analysis there, but then you have to do lots and lots of simulations. So that is another issue of, if one was to take a different point, what would happen? Yeah, and so we haven't done that. It's a good point. Yeah.
Thank you very much again.
Okay, thank you.